This video will go over the tools that we have for working with dot plots within the XelExplorer app. So here we are on the Plots tab. If I want to create a new dot plot, I'm going to click on this plus button, and then I'm going to drag markers over into the plotting area. So I'll start by dragging CD45 into the x-axis, and then CD3 onto the y-axis. This makes our first plot. To make additional plots, I just do the same again. So I'll click on our plus button, and maybe we'll compare CD45 and CD19. To place a gate in either of these plots, I simply left click, hold, release when I'm happy with the size of my gate, and then I'll be asked to name that gate. So we'll call this T cells. I'll be asked whether I want this to be a regular gate or an inverse gate. Regular gate is all the cells inside this rectangle. Inverse gate is all the cells outside of the rectangle. I'll also be asked to color this gate. So maybe I'll make this red. I'll do the same over here. So left click and release. Might call these B cells, regular gate, and maybe I'll make this green. Any gate that you have created can be added to any plot. If I left click in this area, I can add my B cell gate to my first graph, and I can add my T cell gate to my second graph. We have other options within this window. So if I double click on the color of a particular gate, I can change that color. So maybe I will make this instead blue, maybe orange. And you'll notice that any change that I make in one of these plots will update on both sets of plots. I can also change the name of the gate. If I double click on the name, maybe I'll add a space between T and cells, click OK. That will be updated on both plots. I can also remove a gate from this dialog. So if I click on the name or the color of a gate and then press the delete key on the keyboard, I'll be asked if I want to delete that gate. So old T cells old was made just for deleting, so I'll click yes. And now that gate is no longer available. The last thing you can do here is pick a gate and delete all cells that belong to this gate. So this is actually deleting those cells from the analysis, but that is an option that you can do. We also have some options for display that are available by clicking on this options button. So we can close this plot. We can copy this plot to a clipboard and then add that to another application. And we have a few visualization options. First, we can change the size of the dot, displaying the data. We can use auto scaling for X or Y. We can change the size of individual axes. And we can change whether or not we want to display the cell percentages in these quadrants as percentages or as absolute cells. So let's click all these on. Finally, you can also limit the positions that are part of this dot plot. Say, for instance, I wanted to, instead of all positions, look at only positions 1 through 5. And with a semicolon, I would skip position 6 and look at only position 7 through 10. You could save all those changes. And you'll see in this graph, we now have absolute cell numbers being displayed. We've changed the look at the axis, X and Y axis. We've changed the number of cells that are being included in this analysis as well. When you do truncate or limit the number of positions, the positions that are being analyzed will be displayed at the top of the graph. The last thing to cover is that each of these gates is also available for display within the image window. So if we go to image, here we have a portion of those cells. If I go to this options button and click here, I can choose to display my individual gates. So right now the all gate is displayed. We can click that off, and I can choose my T cell gate, or I can choose my B cell gate, both of them. I can also display gates that include cells in two gates, in which case when a cell belongs to more than one gate, it is circled more than once. So in this case, this cell belongs to the L gate and belongs to the B cell gate, so it is double circled.